Okay, great. So here I have my beautiful canvas upon which we'll be drawing. All right, so let's think of a scenario like this. Let's imagine that you have, I'm going to zoom in here. So here's your finger. That's a pretty good finger, isn't it? All right, so there's your finger. And you accidentally are reaching up for something and you poke yourself with a pin right there. So let's write poke. Okay, now with the sufficient amount of force, that poke is going to be felt. You're going to know that you've been poked. And guess how you know that? Well, your brain knows it. But notice how right now we're talking about some stimulus here. So this is the stimulus. That's going to be transferred over to your brain as, some sort of a pain that you want to avoid, right? But you know what's really interesting is that you actually pull away your finger before your brain even recognizes that you've been poked, right? So in this case, we're going to be looking at a reflex arc, an arc that allows that sensory neuron to go right through the central nervous system, but without going necessarily to the brain, but simply going to your spinal cord and then sending that message right back to the muscles to say, oh, we're going to pull that finger away. So that's the type of model that we want to look at today. So we're going to be looking at the stimulus first. So let's do this. Why don't we see what's happening here in this little square and zoom it up a little bit to see how you actually can feel that stimulus, okay? All right, so maybe I can go over a little bit to the right and let's make sure that the paper is aligned properly. So I'm not going to draw like the whole skin stuff, all right? So like, the subcutaneous tissue, like I'm not going to do that. I'm just going to simplify what's happening. So let's imagine that this big boxy looking thing right here, this is the needle, right? Now, of course, if you zoom in on the needle, it's not going to be that sharp. It's going to look a little bit blunt, right? However, we have these cells here on your skin, just like this. And just for the sake of simplicity, let's imagine that you have these sensory nerve cells that are all connected right? Just like this, connected to these skin cells, like so. So right now, what I'm looking at is essentially a cell body here, looking at an axon, right? These are the dendrites. And let's suppose these guys right here are just the skin cells. Okay, so we're oversimplifying what's happening, but hopefully you guys are following what I'm trying to draw here. Does everyone kind of see what I'm trying to draw? I'm not that terrible of an illustrator, am I? Okay, great. All right, so we're going to do this one more time. We're going to have to zoom in just one more time because what I want to do is I want to focus on this part right there, that little circle. Because that little circle is going to sort of get us to the membranous level, right? The level at which we're looking at, things like, channels and pumps and things like that. So let's go ahead and draw that out quickly, shall we? So I'm going to draw out this membrane like this. But as I always say, when I draw things for you guys in my AP Bio class, it's going to take forever to draw every one of those phospholipids. So I'm simply going to, kind of use this line to represent the cell membrane. And let's kind of add a few things here that I think are really important. So one of the things that I'm going to add here is something that you guys are already familiar with. And if I draw something like that with, two different types of substances that can be moved via this pump, hopefully you guys recognize this as being what we call the sodium-potassium pump. All right, that's the sodium-potassium pump. I think you guys all know that, right? So let's put a sodium-potassium pump here. And then what we're going to draw are a couple of channels. And these channels are all important. So the first channel here is going to be what we call a voltage-gated sodium channel. All right. And then I'm going to go down here a little bit. And let's draw a voltage-gated potassium channel. All right, great. So we have sodium-potassium pump, we have voltage-gated sodium channel, we have voltage-gated potassium channel, and here's one more. Now, this is going to be somewhat of a new idea here, but I'm going to draw another channel that looks a little bit different. So I'm going to draw it a little bit fatter here, and this channel is going to be something called a pressure-gated sodium channel. Pressure-gated sodium channel. And I think that should be enough. If I'm forgetting something because it's been years, actually months since I've taught this lesson, then I'll just add it in later. But for right now, let's just focus on these four major membrane-bound players to establish what's going to be happening here in this cell as we look at the scenario. Now, one thing that you guys should know is that the cell above it is probably like this and, has some connections to the glycolipids and so forth, So this is the skin cell up here. And what we're really looking at here is the dendrite of a neuron over here. Okay, great. Now, I know that I've done chapter 5 with all of you guys, so you guys should all be familiar with sodium-potassium pump. But if you forgot, for whatever reason, I'm going to have to explain that first, because sodium-potassium pump plays a critical role in neural transmission. So let's talk about the sodium-potassium pump. If you guys recall, sodium-potassium pump has dual functions. One is that it pumps out sodium, and two is that it pumps in potassium. But the way that it does this is that it pumps out 3 sodium ions for 2 potassium ions that enter. And as we talked about earlier this year, this actually creates what we call an electrochemical gradient. The electrochemical gradient has 2 different aspects, the electrical gradient and the chemical gradient. Let's start with the chemical gradient. One of the things that we can say about this sodium ion is that if we're pumping out sodium ion, obviously, what we're going to be doing is building a concentration gradient wherein the sodium concentration outside is high and the sodium concentration on the inside is low. And another thing that happens because of the potassium being pumped in is that the relative concentration of potassium is going to be high and the relative concentration of potassium is going to be low on the inside and outside of the cell. So this is something that we've discussed earlier. But there is one more thing that's happening, and that is that because we're pumping out 3 positive ions, while only pumping in 2 positive ions, we have a slight negativity that we call the resting membrane potential on the inner side of the membrane. So the outside of the membrane becomes slightly positive, the inside of the membrane becomes slightly negative. We call that the RMP, the resting membrane potential. And if you guys recall, we can sort of track how this resting membrane potential behaves by simply drawing, let's say 0 millivolts here, and maybe somewhere around minus 50 millivolts here at the bottom, and the RMP would be steady at about minus 50 to minus 55 millivolts. That's the standard typically for the resting membrane potential of a cell. So up to this point, this all should have been a review. So far, so good? Great. All right. Now, how do we get the information of this pinprick into this nervous tissue, into this neuron? Well, let's take a look at the bigger picture here. As the needle presses down against this cell is going to press down against the neuron too, because, well, physics can't really escape it. So if we kind of looked at the zoomed in section, as the skin cell starts to press down against this neuron, it's going to do something to this guy on the very right side, the pressure-gated sodium channel. Now remember, this pressure-gated sodium channel is a channel that can open or close. And if it's open, it'll allow sodium to flux. If it closes, it won't allow sodium to move. But how is it gated? It's gated by pressure. Now let's think about what's going to happen. If this skin cell gets pushed down, it's going to push down against this protein right here. And it's going to create pressure or a distortion. And remember, one of the most wonderful things about proteins is that these proteins have the capacity to change shape and respond in response to things like chemical changes, which is something that we're familiar with, but also mechanical changes too. So this is a mechanically gated sodium channel. And as the mechanics of the pressure exerts some level of distortion on this channel, it opens. All right. So the pinprick happens, the skin cells get pushed down, distorts the protein opens the sodium channel. What is going to happen to the sodium ions? Look at the diffusion gradient. Sodium is higher on the outside, lower on the inside because of the sodium-potassium pump. So through this sodium channel, we have sodium which begins to enter the cell, just like this. Does that make sense to you guys? Sodium begins to enter the cell. All right. Now, this doesn't necessarily mean that we have a signal, but going back to this little drawing here, you could imagine that if that pressure-gated sodium channel is open and sodium is coming into the cell, you're going to start to get the cell to sort of become a little bit more positive because you got positive ions coming in. Now, it's not positive. It's still negative, but it's starting to become a little bit more positive. Now, here's the thing. If this pinprick was so subtle that this skin cell went right back up and you kind of just simply grazed over it, well, then you could imagine that this channel will close, yeah? And if that channel closes and this sodium never really gets that high, then, you know, it sort of goes back down to the resting membrane potential and nothing happens. And you know what? You would never even know that this happened because a signal was never sent to your brain. However, that's kind of boring, right? We want to know what happens if you kind of sustain that pinprick and, you know, hold it to the degree that it causes your brain to react. So what we're going to be doing is we're going to keep the sodium open a little bit longer. And as we keep it longer, sodium comes in and in and in and in and in until you reach, what happened? Sorry. Until you reach this point right there, which is called the threshold. The threshold is the point at which the cell becomes sufficiently positive that there's no return at this point. You're going to send that signal. And the reason at the cellular level that this is in fact a threshold is that whatever voltage this is going to be, let's just say that this is minus 30 millivolts. This minus 30 millivolts is the voltage at which all of the voltage-gated sodium channels open. And by the way, I've only drawn one voltage-gated sodium channel. There are hundreds of them, and they all open at the same time. Okay, so let me rephrase that because it's kind of hard to imagine from what I've drawn in the picture. So you have the pressure-gated channel that's open because there's pressure on it, and it allows sodium to come in, and that sort of builds up. But if it builds up to a sufficient degree and your cell decides, you know what, this is a problem, we need to send a signal, well, as soon as it reaches that threshold potential, all of the membrane-bound voltage-gated sodium channels will open simultaneously in this local region. And what that means is a crap ton of sodium is going to come in all at the same time, right? Because it's not just this one, there's hundreds of them. So what would that look like? That would look something like this. That would look something like that. All the sodium pumping into, flowing into the cell would make this cell no longer negative, but very positive. Now, this portion right here, past the threshold right here, this is what we call depolarization. Depolarization. It means that the cell is suddenly becoming very, very positive. Is everyone following so far? That, ladies and gentlemen, is the point at which this neuron now has received the electrical signal. The electrical signal in a body is through the flow of positive ions, the sodium ion. Now, the electricity that we use in, your electronics is actually electrons. Here, we're talking about positive ions. So it's actually the opposite, but you could imagine electrical current is a current, right? So here we have the depolarization. Now, I'm going to just go ahead and talk about what happens at the peak of this depolarization, and then we'll kind of step back and think about how this actually works with respect to the whole membrane of the cell. But once you get to this point, we reach the peak, and maybe this is something like plus 35 millivolts. I'd have to double check the numbers here. But that is when the voltage-gated potassium channels open. Now remember, what happens to potassium? Well, remember, the concentration of potassium is actually higher on the inside than on the outside. So this is called a peak because once that potassium channel is open, the sodium channels close. And only thing that happens is for the potassium to now flow out of the cell. Remember, potassium are positively charged ions leaving the cell to the exterior, making that cell now more negative, dropping that voltage back down. Because potassium is now leaving. And then it returns to its normal state after a little bit of what we call hyperpolarization, Why? Well, that's because potassium channels don't really close exactly at the resting membrane potential. They actually close slightly under it. So it goes a little bit of an overshoot downwards, and then it returns back to normal. How does it return back to normal? Well, that's because the sodium potassium pump is, again, regulating the concentration of sodium potassium in general, right, on the background anyway, whether this is happening or not. So what we've looked at is what we call a single action potential. A single action potential that allowed us to sort of understand how the initial sodium influx began with that pressure gated channel, and then how the slow rise in the sodium concentration made the cell positive to the degree that voltage gated sodium channels all opened and allowed a lot of sodium to come in, causing a depolarization. And then we learned that at the peak of the action potential, how the sodium channels close and the potassium channels open instead, allowing that cell to repolarize again. And then we learned that the potassium channels are into a little bit of a hyperpolarization, ultimately returning back to normal again at resting membrane potential with the assistance of the sodium potassium pump. This is a single action potential. We haven't really done much though, because as far as you guys can tell, all we've done is just sort of made this little local region. So if I may use a different color to sort of designate what's happening. All we've done, as far as you guys can see, is that we've made this part of the cell a little bit negative, right? Or a little bit positive. Sorry, we've just made this part of the cell a little bit positive. But here's the important thing. So let me go back to black. So one of the things that happens is that, remember that this cell is actually super, big, right? It looks something like this, So here's a dendrite. So what happens is that as this sodium diffuses, it allows voltage-gated sodium channels to open here, boom. And then as the sodium comes in, it allows voltage-gated sodium channel to open here, boom, 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 boom. Do you guys kind of understand what I'm trying to draw here, right? It spreads. That signal spreads. Why? Well, diffusion. So as the sodium diffuses to the nearby local region, it allows the next round of voltage-gated channels to open. So more sodium comes in. That sodium diffuses here, opens this voltage-gated channel, so it comes in. And one of the things that you'll notice is that this signal is going to go boom, 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 and boom, and boom. It travels along the dendrites all the way to, guess where? The hillock. The axon hillock. And if at the axon hillock, the signal is still strong enough, it's going to do the same thing by spreading through the axon. And if you look at the axon, I'm going to draw an axon right here. The axon literally is just simply channels like this. So as the sodium moves in this way, opens this channel, more sodium comes in. And they move this way, this channel opens, this channel opens, this channel opens. And by the way, one of the cool things is as the sodium influx continues to move this way, it closes the ones behind it. So it's like plus, plus, minus, 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 right? And then it becomes plus here, this becomes minus, minus, plus, plus, minus, minus. I know I can't really demonstrate that very well on a whiteboard, but I think you guys get the idea. It's almost like if you were to watch, like a wick and it's connected to a dynamite and you put the flame here, it goes tss, right? That's kind of like what's happening. So it travels down the axon like so, boom, right? Dun, 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 causing that action potential again and again and again and again until it reaches our terminal. Okay. Now I did promise that we're going to actually get to a point where this finger gets actually, taken aback. So let's actually see what's happening at a synapse. Because right now we didn't look at a synapse, right? We looked at just the area at which the sensory input is. And then we've gotten to the point where we're understanding how that electrical signal or the positive ion, positive influx, that's the electrical signal, by the way, the positive influx of sodium is traveling down the axon. But we haven't really talked about what happens at the axon terminal. So imagine that we go like this and we finally get to the axon terminal, which is right here. You know what? I actually want more space. So can I just erase this, guys? And let's just redraw what's happening at the end of the axon terminal. So I'm just going to simply have this axon come out like this. And then I'm going to say this is the synaptic So this is what we call the axon terminal. And let's have another neuron here. So this might be an interneuron, but let's have a neuron here. And as you guys remember, this area is called a synapse. And what we're going to be focusing on is this area right there. Okay, so let me try to draw that out in a slightly greater detail here for you guys. All right, so let's draw that a little bit bigger. So here I'm going to draw the presynaptic neuron. And here I'll draw the postsynaptic neuron fairly largely. As you can imagine, here we have voltage-gated sodium channels, right? You guys know that. So here, voltage-gated sodium channels. So as the sodium diffuses down, so let's imagine that that sodium diffuses down this way, it creates a positive depolarization, which then opens these channels and allows more sodium to come in. And that's basically what we said happened with the movement of the electrical signal. All right, so a couple of things to introduce here. As you guys remember, this synapse is not connected together electrically, but it's rather connected together chemically. And those chemicals that we release from the presynaptic neuron here are going to be called our neurotransmitters. Neurotransmitters. So where are those neurotransmitters? They're in vesicles, guys. So we have all of these vesicles that are ready to be launched into that synaptic space. And let's go ahead and draw these neurotransmitters in pink. Now, if anyone were to ask you, well, how do you guys make neurotransmitters? Yeah, you put them into the endoplasmic reticulum and then you create these vesicles to the endomembrane system, right, guys? So this is all connected to everything that we've been doing in the cell biology unit as well. All right, so here's what happens. So these are voltage-gated sodium channels. Okay, so I'm going to write voltage-gated sodium channels here. But at the end of the presynaptic neuron, we actually have another type of voltage-gated ion channels. So I'm going to draw it like this. And this one, I'll just draw one here. This one is actually a voltage-gated calcium channel. Calcium. Now, remember in chapter 11, I said, yo, guys, calcium is responsible for a lot of cell regulatory mechanisms. Remember I told you that? This is why we always pumped calcium out, and this is why calcium was pumped into the ER, and we always had a low cytoplasmic calcium concentration. Hopefully you guys remember that. So guess what? As these sodium ions come into the synaptic cleft, they create a depolarization, a positive, right, positive membrane potential, which then triggers this voltage-gated calcium channels to open. And because we've already established that the concentration of calcium is always high on the outside with respect to on the inside, calcium is going to come in. So calcium is going to come in. Now, we don't have to understand exactly how calcium does what it does, but it facilitates the merger of these vesicles to the synaptic cleft so that you can kind of see what's happening, right? What calcium does is it allows those neurotransmitters to be released into our synapse. Okay, so we're just kind of going everything through the, like step by step mechanically, but let's just review that because this part is a little bit tricky. So through the axon, we have the electrical signal that's coming down because of obviously our voltage-gated sodium channels. But at the end of the neuron, instead of opening additional voltage-gated sodium channels, because we have to transfer this electric signal to a chemical signal, we instead open what we call voltage-gated calcium channels. And when calcium comes in, it acts as a secondary messenger, which then binds to these vesicles and allows those vesicles to merge with the membrane, allowing these neurotransmitters to be released into our synapse. Okay, great. So we're almost there, guys. We're almost 90% in being able to transmit this information to the postsynaptic cell. So here, let's write our postsynaptic cell. Okay, now, I didn't really draw everything that I needed on the postsynaptic cell, so let me just take a moment to do that. So I'm just going to draw a few. All right, so here we go. Boom, boom. You know what? I need a little bit more space for these guys. So let me draw it like this. I'm just going to take some time drawing this because this guy is kind of cool. Like, looks like Rudolph. I don't mean to draw it like Rudolph, but All right, here we go. So let me label some of these guys too. All right, so these Rudolph-like looking things right here, this is called a ligand-gated sodium channel. And this is ligand-gated sodium channel. Voltage-gated sodium channel. Voltage-gated sodium channel here. By the way, they don't have to be necessarily in this order, right, guys? Voltage-gated potassium channel. And let's do one more voltage-gated potassium channel. So I think you guys know where I'm going with this. Take a look at this. Ligand-gated sodium channel are proteins that you know that can open or close the channel based on ligands. Guess what the ligand is? That's the neurotransmitter, guys. So as the neurotransmitter binds, boom, 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 open, 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 open. What comes into the cell? Sodium starts to come into the cell. What does sodium do? Sodium starts to make the cell a little bit positive. What does that do? It activates the voltage-gated sodium channels all at once if sufficient amount of sodium comes in. And voila, all the sodium comes in. Boom, 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 boom. What happens to this cell? This cell becomes depolarized. And at the peak, sodium channels close, potassium channels open, potassium leaves, hyperpolarizing back to normal. This is how that action potential gets transferred from the presynaptic cell through the chemical to the postsynaptic cell. It's amazing how this happens. But you see, the thing is, on this postsynaptic cell, instead of the original depolarization coming from the pressure-gated sodium channel that we talked about, it's simply happening as a result of the ligand-gated sodium channels here. And whatever the neurotransmitters may be, acetylcholine, GABA, whatever, you're going to have a very specific neurotransmitter that's able to attach to a very specific sodium channel that opens that channel and transforms that next postsynaptic cell again into the depolarized state, which then can carry on the message, All right, so let's imagine that this is a cell that simply says, oh no, you've touched something bad. Let's just go ahead and take this and connect it right to the muscles. Oh, actually, you guys know that there are no muscles in your fingers, right? The muscles are actually in your in your forearms. The muscles that move your fingers are all on your forearms. There's only tendons and ligaments here. And you know, if you think about it, it makes sense. You know how if you work out a lot, your muscles get really big? If you had muscles in your hands, which we use all the time, your hands would become so sausage-like that you would lose all articulation. So evolution, as it would have it, placed all the muscles that move your fingers in your forearms. So this neuron, I know that was completely unnecessary, but hey, might be useful on trivia night. This neuron goes into those muscles, boom, 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 says, hey, contract. Move that finger away from that pin. And we didn't talk about that portion yet, right? Because we're going to talk about muscular contractions later. But boom, that contracts, move your finger, and there we go. We have a reflex arc. And we actually can understand the whole thing by looking at every single mechanical aspect of this. The only thing that we kind of cheaped out on, guys, is how that calcium helps with the exocytosis of the neurotransmitter. But other than that, you know, we're pretty good at this. Like we looked at everything. So I wanted to use that example because I feel like it's something that most of you guys had experiences with. It sort of covers the idea of the peripheral nervous system, the reflex arc, and how the muscle can be contracted. And of course, it goes into the details of how all of this depolarization stuff works. So I feel like you guys have a much better idea of now what's happening in this picture. Let's see how the book explains what's happening. So let's take a look here. We got the sodium-potassium pump. We got the potassium channel. We got the sodium channel. Inside the cell, you have a lot more sodium. Outside the cell, you have a lot more potassium. But overall, you can see that, yes, there is the chemical gradient, but we also have our electrical gradient. So the resting membrane potential they talk about here goes to showing, in this picture, is showing about minus 60 millivolts, but that's okay. Minus 60 millivolts in this picture. It's showing you that action potential right here. And it's showing the threshold as being about minus 55. I think that is a little bit low. But it depends on the type of cells. So, you know, we'll take it as it is for this chapter. But, you know, the way that this book does it is a bit confusing. So they kind of show it like this, right? The role of voltage-gated ion channels in the generation of an action potential. They show you the threshold. And then Number 3 here is showing you all of the voltage-gated sodium channels opening, all the sodium coming in. And then you can see the falling phase, which is in green here, showing you that the sodium channels are closed, but the potassium channels are open. So it's allowing all the potassium to flow out and so on. So this is not really anything new. So this is fine. There is one thing that I like here, and it's this picture. This is the picture that I was trying to draw for you guys as how the action potential travels as the sodium kind of goes in, but how the, axon that's being left behind, right, so to say, repolarizes and becomes normal again. So it's almost like a traveling wick with that spark traveling down the axon. So there's one more thing that we need to add here before we look at the finer control mechanisms of the nervous system, and that has to do with something called myelin sheaths. So when you take a look at an axon, it actually isn't fully exposed like the picture that we saw. It actually looks something like this, with all of these insulation that's being provided by what we call the Schwann cells. Now, the Schwann cells are actually cells of the nervous system that are supposed to help, that are support cells for the nervous system. And what they do is, as they develop, they sort of wrap around the axon almost like insulation. Almost? No. They're exactly like insulation, because what happens with the myelin, which is what we call them, the myelin sheaths, what happens is that it blocks all of the sodium channels. Now, why is this important? Well, going back to this picture, you could imagine in a perfect world, this signal will never die out. But in the real world, you have what we call leaky channels and leaky membranes, as in the membrane is not always completely impermeable to sodium and potassium. So what that means is, while we want a lot of sodium to be coming in, sodium leaks out. And over large distances, sufficient number of sodium ions can leak out and potassium can leak back in to kill the signal in its step. So what would happen? You would have basically paralysis. So what we do here is by using myelin, okay, what we call myelination. Through myelination, we're using Schwann cells to wrap around and ensure that no ions can either enter or leave through the length, or at least certain lengths of the axon. So where the resurge of depolarization occurs is really only at these points between the Schwann cells that we call the nodes of Ranvier. Nodes of Ranvier. So here's a better picture of this. As you can see, we have what we call a saltatory action. Now, saltatory means jumping. We have this jumping action, as in depolarization occurs here, boom, and then boom, and then boom, boom, and boom at each of that nodes of Ranvier, where fresh sodium ions will come in and will accelerate towards the other side, and boom, new sodium ions will come into the voltage-gated channels, and it will just kind of jump from place to place. So this idea of saltatory action of the nervous system is something that you have to be very familiar with, and you should know what myelin is and what Schwann cells do for SAT biology and as well for your general livelihood. One thing that I should mention here is that the death of myelin sheaths, or the death of these Schwann cells, are I guess, thought to be responsible for some neurodegenerative disorders. So, for example, Lou Gehrig's disease, or ALS, amyotrophic, I forgot what the name is, but multiple sclerosis, lateral sclerosis, right? Amyotrophic lateral sclerosis. So these are diseases that are associated with the death of the Schwann cells, therefore not having that insulation, therefore the cells not conducting the signal properly and the signal dying halfway through, therefore leading to things like paralysis. Right, guys? So this is very important that we have proper myelination. Now, going to the synapse and how your book covers the synapse, they tell you, okay, so you have these neurotransmitters that are released as a result of calcium channels opening. Calcium comes in, allows for exocytosis. These bind to additional channels, ligand-gated ion channels. The idea is, whether you have sodium and potassium or just sodium, you know that potassium will leave and sodium will come in, which is all good, which is all what you want to happen anyway. All right? So that is how that signal gets transmitted to the next stage. So what we're going to be talking about now is, I guess, looking at this from just sort of a broader perspective and thinking about, how these signals can actually be calculated and integrated within the ganglion, and not even just at the ganglion, but at junctions that we call interneurons. So the places, so the nerves between, the neurons between neurons that have the capacity to make decisions. So, for example, we've been talking about just this one connection right here, where the presynaptic neuron connects to, say, suppose, I don't know, the ganglion, the dendrite or the cell body of a next neuron. But in reality, you don't just have one neuron connected to the next neuron, you have several neurons that are connected. And what's even more fascinating is that certain neurotransmitters being released by what we call excitatory signals are going to make it more likely for the action potential to be had in that postsynaptic neuron. But there are also other neurotransmitters that create inhibitory responses too. Inhibitory responses make it less likely that an action potential will occur. How would we do that? Essentially, it would release a neurotransmitter that allows for not the sodium channels to open, but rather the potassium channels to open. Because if you think about a neurotransmitter that controls a ligand-gated potassium channel, as soon as it binds, the potassium channel opening will allow more potassium to leave, making that cell more negative. The more negative the cell gets, the harder it is to get it to the threshold where you have the action potential that's actually going to take place. So you have many possibilities here, and we have 4 examples of what could potentially happen. And one of the things that we're going to be looking at is right here, where you have 2 signals that are coming in, but are coming in at such a distance in time scale that they never really add up to meet that threshold. So this is kind of like what I said, when you have your finger on the pin, but then you so slightly graze over it that everything closes up and you don't have sufficient sodium coming in to open the voltage-gated sodium channels. So if you only have one neuron that's touching it, or not touching it, but it's giving it a positive signal, but it's happening across a longer span of time, then it allows for that cell to return to the resting potential, which means no action potential will occur. But the first thing that we're going to look at is what happens if we really want that signal to go. Well, we have something called temporal summation, as in you have not one signal coming in, but as soon as that signal gets transferred, you have another signal that comes in. So before the cell can return to its resting membrane potential, another influx of sodium ions will push it above and beyond the threshold point where the action potential will take place. So this would be like duk-duk, right after each other. Now, of course, that would cause an action potential, but so could 2 signals that are simultaneously coming in on both the excitatory front. So for example, in this case, we have what we call a spatial summation, as in 2 different spaces, 2 different areas of that cell body, you have sodium ions coming in, driving that membrane potential to a sufficient, depolarized state that opens all the voltage-gated sodium channels, allowing that action potential to happen yet again. And you can see that the grading at the axon hillock here is deciding that the signal will go. The third one is what we call a spatial summation of excitatory presynaptic potential and inhibitory presynaptic potentials. That's what EPSP and IPSP means. But in this scenario, you can see that, yes, you have the excitatory signal coming in, but then at the same time, you have an inhibitory signal coming in. And those things would sort of kind of even out and sort of kill that wave, almost like noise-canceling headphones. Like if you guys know a little bit about physics, noise-canceling headphones work as a result of producing a sound wave that's exactly opposite to the sound that's coming in from the outside, therefore canceling out those waves. That's essentially what's happening here, almost canceling out in this case, the voltage to make sure that nothing will happen. So at this point, you might be wondering, well, why would we want an inhibition instead of excitation? Well, the thing is, life isn't really that simple, right? You have to make decisions that are really, really complicated, even at the level of a couple of neurons. So knowing whether you're going to do something or not is going to be contingent on numerous sets of information that are simultaneously being graded. So this is why we call this the summation, or sometimes the grading of signals, so that the cells can determine whether it's actually going to send that signal or not. Now, what's really fascinating about all of this that we're talking about right now is that your brain is literally trillions and trillions of neural connections that is basically bringing in all the information from every single sensory part of your body, and feeding it through this large, massive circuit of connections that are simply on or off, and then eventually deciding what to do with it. And somewhere between the sensation and the output lies what we call our consciousness. And the amazing thing about consciousness is that it's an emergent property that arises as a result of the complexity of the neurons that we have in our brain. And we really don't know anything about it. Like, for instance, we know that some animals with sufficient degree of complexity in their brains have consciousness. If you guys have dogs or cats, you know that they are conscious beings and they can feel and they can behave and they have moods about them. But then to what degree do we give that privilege of consciousness when we look at lower and lower and lower organisms, like grasshoppers, for example? They certainly have brains or ganglion. Worms have ganglion. You see, we don't really know. And the thing is, we won't know for a long time. And I think this, in some way, is what's really inspiring about this type of study, is that when you study AP biology, we tend to study a lot of what people discovered in the 1800s and 1900s. And you sort of feel like, well, why am I just looking at things that have already been discovered? But as soon as you kind of start to go a little bit beyond what we've learned, you start to realize that there are still so many unanswered questions. And that is exactly why science exists. And that's precisely why I'm sure you guys are actually here with me a couple of days after Christmas and a couple of days before New Year's studying this, because it is very interesting and we really do want to know what the hell is going on. So it's a really cool thing. All right. In any case, that's a little bit of a pep talk to keep you guys paying attention. But the point is, yes, this sort of interactions between the synapses and between the neurons is essentially what allows every organism to behave in a certain way. All right, now, neurotransmitters. So let's focus a little bit on neurotransmitters. Oh, by the way, I forgot to say something very important. One of the reasons why this also has to exist, and this is something that you guys should definitely know for subject test biology, is that action potential is an all or none response, right? As you can see right here, we either don't have an action potential or we have an action potential. And that is a bit of a problem, because if you're looking at a single neuron that can either be on or off, then it makes things way too simplistic for difficult decisions to be made, which is precisely why multiple on or off systems are connected together to determine the next system's on or off, which then connects to other system to make sure that they can either turn on or off through a complex network of things that are either on or off. Does this remind you guys of anything? Computers, binary system, either ones or zeros, right? Same thing. Computers either have a on or off system, a language through which they can make very, very complicated decisions. And that's exactly what's happening here too. We've actually kind of modeled computers after ourselves, even though we didn't really know it at the time. That's kind of cool. Neurotransmitters, ladies and gentlemen. So this is not as important, to be completely frank with you guys, but there are a couple that I think you guys need to know. Acetylcholine, that you should know. It's a major neurotransmitter that is found at the neuromuscular junction. So if you recall that picture that we drew, we said that there was going to be a neuron that connects to a muscle in your forearm that allows your finger to twitch. That neurotransmitter that's being released between the presynaptic, well, it wouldn't be a presynaptic, but the final motor neuron and the motor unit, which is our muscles, is going to be acetylcholine. But we also have things like biogenic amines, amino acids, and neuropeptides and gases. And I'll just show you a brief picture of what they look like and where they're used, just focusing on a couple that I think you guys should know. Acetylcholine, as I said, very important for skeletal muscles. They can be excitatory or inhibitory at other sites, but excitatory at the muscles only. And it's secreted by the central nervous system as well as the peripheral nervous system at neuromuscular junctions. Biogenic amines, you probably are familiar with most of these guys. Norepinephrine, or what we call noradrenaline, is a hormone that actually acts as a neurotransmitter, which is kind of cool. And remember, this is in the fight or flight response. Dopamine, as you guys know, dopamine is going to be your feel-good hormone, in this case, acting as a neurotransmitter at the central nervous system typically. And dopamine here obviously makes you feel awesome, which is why many drugs that are typically what we call the opiates, they mirror the dopamine structure to get those dopamine sites activated to make you feel awesome. But don't do drugs because, you know, your body will become dependent on them, particularly the ones that mess with your neurotransmitters. Serotonin, okay, generally inhibitory, not to be confused with melatonin. So serotonin, I think, regulates your mood in the central nervous system. As you can see, that's going to be your brain. We also have amino acids like GABA, which is gamma aminobutyric acid. And we also have glutamate and glycine. All glycine, just straight up glycine. You guys probably know what that is already. Glycine is going to be that very, very simple amino acid. They all play some role in the neurotransmitter industry, but you don't have to really know too much about them. Substance P, met, and caffeine, you don't really have to know. Nitric oxide, this one, kind of interesting. Nitric oxide is the only one out of the neurotransmitters that are actually gaseous, which means that it diffuses straight through your blood vessels into these junctions. But the interesting thing about nitric oxide is that it's typically used in the vasodilation, as in it dilates your blood vessels, which is one of the reasons that nitric oxide was initially used to treat things like heart attacks, because remember, in heart attacks, one of your coronary arteries gets blocked up. You want vasodilation so that those arteries open so that you can get blood flow. And then what they found out was that the same, vasodilating effect that it had on the heart also kind of transcended to the male reproductive parts and also allowed for erectile dysfunction to be mitigated, which is why nitric oxide is the primary constituent of Viagra, if you guys know what that is. And that is, kind of an interesting fact. Anyway, not much more to say about that. And we don't really have to talk about the rest of this stuff. So I think at this point, we could really move on to the muscular structures and kind of link it to what we've been dealing with here with the neurology of things.